Okay, next I want to talk about eukaryotic genomes. And again, these are mixed genomes because there have been a lot of endosymbiotic events going on. So they're more complex, they're generally larger, and so I'll talk in a more general sense about these genomes. So here are some of the genome sizes of eukaryotes on top. You can see the protozoans on the bottom, the protists. And you can see it's a broad range, and this is because that includes all the way from excavata and the rhizaria, acrobians, alveolata, stromatophiles, etc. So it's a wide range of organisms. Plus, these ones, remember, have picked up, many of them at least, have picked up a eukaryotic organelle within them. So they have larger genomes, having included at least two major eukaryotic genomes. And then up into the plants, quite a broad range in those. Again, the black indicates those that are free-living. The grayish color indicates a mixture of free-living and parasites, and then the white are parasites, obligate parasites, etc. So plants, you can see going from the algae, mosses, lycophytes, monilophytes, gymnosperms, and angiosperms there, and then up into the Opisthokonta, which includes the fungi and the animals, going from invertebrates up to mammals. Also note in this one that I've included dinosaurs. The way you get dinosaur genome sizes is in some fossils you can see cellular structure, including visualization of the nuclei. And a nucleus volume is proportional to the genome size. And so this group of researchers then determined sizes for stegosaurs and other dinosaurs. And interestingly enough, they match up pretty well with reptiles and also with birds. And birds are one of the sister groups to dinosaurs that branched off a couple of 100 million years ago. So here on this tree are the eukaryotes, including kinetoplastid diplomonads. Those are the excavata, then into the chromalveolates, which are the stromatophiles, alveolates, and other protists, plants, amoebae, fungi, and animals. And fungi and animals are within the Opisthokonta. So here we focused in a little bit, so I'll talk about a couple of different genomes within this group. So the first one I'll talk about is Arabidopsis thaliana, a small little plant within the cruciferae. And then another group of plants I'll talk about. And then I'll speak on the fungal Saccharomyces cerevisiae genome, as well as a couple of animal genomes. Arabidopsis thaliana was the first plant that was fully sequenced, and that's because it has a relatively small genome. This plant it has been studied for many decades now, and that's primarily because it's small, has a very fast life cycle, and many mutants have been produced, so that it's been studied, dissected essentially developmentally in many different ways. And we talked about this one a little bit before when we talked about development in plants. So here's a pie chart of the Arabidopsis thaliana genome. It has 157 million base pairs of DNA in its genome. Now that compares with about 12 million bases for Saccharomyces cerevisiae and about 3.2 or so billion base pairs in humans. It has between 27,500 and 31,000 protein encoding genes, and is able to produce about 35,500 different proteins. So there is some alternative splicing going on in Arabidopsis, but it's a much smaller amount of alternative splicing than you see in animals, for instance. Because its genome is so much larger than things like Saccharomyces, then the percent of the genome set aside for RNA and DNA metabolism, signal transduction, cell organization, transcription, etc., appear to be smaller, but it actually has more genes involved in those functions than in Saccharomyces cerevisiae. But it has quite a few more genes involved in developmental processes, since it is a large, relatively large multicellular organism. And a lot of its genome is devoted to responses to stress and response to external stimuli. I mean, plants are out in the sun, they're out in the cold, they're out in the heat, they're out when it's wet, when it's dry. So they have to respond to a large number of environmental conditions. So they have to be able to switch on and off different genes to respond to those. They're also attacked by pathogens constantly, so they have to react to those pathogens as well. So they have response mechanisms either to avoid pathogens or to wall off certain cells that are being invaded. Here's a look at what is on each one of the chromosomes. So you can see that the genes are pretty much spread among the 5 chromosomes, although the largest one is chromosome number 1 with a length of 30 megabases, which is about almost 3 times as large as the entire Saccharomyces cerevisiae genome. And then there are the number of genes and the number of proteins made. So you can see the number of proteins made is larger than the number of genes, which indicates that there is alternative splicing going on. So you can get more than one protein formed off of a single transcript. Also, there are 570 to 750 ribosomal RNA genes. And we discussed earlier in the semester why you have so many ribosomal RNA genes in plants, and then they're quite variable. So even in a single plant, you can have from 570 to 750 on a chromosome, and that you get recombination occurring, and it's unequal, and so one chromosome gets more copies, one gets fewer. Probably the minimum number of genes that an individual cell needs would probably be in the range of 200 to 300 ribosomal RNA genes. Then there are 689 tRNA genes, at least 70 SNO RNA genes, that is small nucleolus organizer genes, RNA genes, and then 177 microRNA genes. And then these then control both the processing of ribosomal RNA genes for SNO RNAs and control mRNA expression in the case of microRNAs. So these would be in very low number in other types of organisms. But by the time you get to multicellular organisms, plants and animals, then these small RNAs really increase greatly in number. So as you know, when there are many different locations in the cell and many membrane-bound organelles within a cell, you can have proteins winding up in different locations. So this pie chart shows the destinations for those proteins. So about 8, almost 9% go to chloroplasts, and 3.4 to go to plastids in general, including amyloplasts and others. Then a number go to the nucleus, to the mitochondria, to the plasma membrane, other membranes, and then to various other locations such as the cytosol, ribosomes, etc. So these are where those proteins wind up in the cell. So although in bacteria, and especially in planktomycetes, you do have signal peptides that take proteins to certain locations in a cell, in the case of higher organisms such as plants and animals and protists, where you have many different cellular locations and many different membranes, there are much larger numbers of signal peptides that aid in getting those proteins to the right location in the cell. The next genome I want to talk about is Oryza sativa, which is rice, and there are many varieties of rice. The genome size ranges from around 380 million base pairs to over 400 million base pairs, depending on which variety of rice has been sequenced. Remember last time I talked about Arabidopsis thaliana, and its genome size was about 157 million base pairs. Both of these actually have fairly small genomes for plants, and that's one reason why they were the first ones sequenced. Rice, as you know, is an important food crop. It's a staple in the human diet for about 3.5 billion people in the world. And if you count rice, wheat, and maize, they provide approximately 40 to 50% of the calories consumed in the world by humans. So it's very important. It's quite varied. So people have grown rice for many, many thousands of years, probably 10,000 years when it was first domesticated. And now there are hundreds of varieties of rice, including long grain, short grain, some that have more starch, less starch, etc. Here's the karyotype and genotype, essentially, of Oryza sativa. And you can see it has 12 chromosomes and then a mitochondrial genome and a chloroplast genome. Each of the chromosomes has between about 23 million to 45 million base pairs of DNA. And again, that's between 2 and 4 times the size of the entire genome of Saccharomyces cerevisiae, just for comparison. Each of the chromosomes has several thousand genes on those chromosomes, and you'll note over here that the amount of alternative splicing is rather low in rice. So there is a bit of alternative splicing, but pretty much it's one gene makes one protein. However, if you look at the total number of genes, there are 61 to 62,000, and that's about the number of proteins it can make. So a large number of genes and a large number of proteins. If you look at the human genome has about 23,000 to 25,000 genes, but it encodes for about 90,000 proteins. So because of alternative splicing, there's a different strategy, genomic strategy, I guess, for producing more genes. One is using each gene to encode a single protein, and the other one is using genes sometimes to encode for multiple proteins. So in general, this is what you find when you compare plants with animals. Plants usually have more genes, but they are able to encode for fewer proteins than in animals, because animals utilize alternative splicing to a much greater extent than do plants. If you compare the rice genome to other groups within the Gramineae, the grass family, you find that they have increasing amounts of DNA, whereas the number of genes doesn't really change that much. The organisms that I want to compare as far as their genomes in the grass family are rice, millet, sugarcane, sorghum, maize, wheat, and oats. And that's going from smallest genome to the largest genome. Here are a few photos of those plants with their genomes. As I already said, rice ranges from about 380 million base pairs to well over 400 million base pairs. There's millet at 490 million base pairs, sugarcane at 1 gigabase of DNA, sorghum at 1.6 gigabases, maize at 2.5 gigabases, wheat at 6 gigabases, and oats at about 11.3 gigabases, which is much larger than the human genome, right? Which is at about 3.2 to 3.5 gigabases. You'll note also that the genome size doesn't really relate to the size of the plant. So rice is rather short, millet's sort of short. Sugarcane is quite large, as is sorghum. Maize is of moderate height. Wheat can be from very short to maybe 5 or 6 feet tall for the taller varieties, and oats also are similar in size to wheat varieties. These are the chromosomes that are lined up in each one of for each one of the grass species that I was talking about. And they're lined up so that you have chromosome synteny. That is, the same genes are lined up in each of the chromosome sets. So in the center you have the smallest genome, that's rice, which has 12 chromosomes. And then you move out, and the genes that are syntenous with those are lined up, and so the order of the chromosomes is slightly different. But in millet there are 9 chromosomes, and you can see them there. Moving out to sugarcane, it has 12 chromosomes, and even though one is rather short and there's quite a deletion in one place. And then moving out further is sorghum, and that has 10 chromosomes again. So you can see where some have split chromosomes to have more chromosomes, and others have joined pieces of chromosomes together to make larger chromosomes. Then you move out to maize, which is a little more complex in that you can see 2 sets of chromosomes, but it really has just 10 chromosomes. There has been an ancient duplication of the chromosomes, so you get 2 sets of those chromosomes lined up in different places and in slightly different orders, so the chromosomes look different in those ones. Moving out to the next ring, those are the chromosomes for wheat, but that's only the haploid of one of the genotypes that have been brought together to make wheat. So there are some diploid wheats, but then there are some tetraploid wheats, and then there are hexaploid wheats, so that bread wheat that you use is hexaploid, whereas wheat to make noodles, durum wheat, is a tetraploid. So they have different sets of chromosomes. And these different sets of chromosomes then bring in different properties. So you have some wheat that has a higher sugar content, other wheat that has a higher starch content, etc. But you can see that this is just one genotype. Actually, the 3 genotypes came from 3 different species of grasses to make Triticum aestivum. So they have 3 very separate genotypes, an A type, B type, and D type. And as I said, the haploid number of chromosomes in wheat for the diploid is 7, and so for the diploid it's 14, and by the time you get to the hexaploid, there are 42 chromosomes in the hexaploid wheat, and 14 of those came from one species, 14 came from a different species, and 14 came from a third species, all having the same chromosome number but being different grasses. And you can see from the diagram that there's been quite a bit of rearrangement of those genes on those chromosomes compared to the other grasses. And then finally out to oats. Oats are another species, cultivated species that is hexaploid. So this is just showing a haploid version of one of the 3 genomes that came together to produce oats. And again, you can see a lot of rearrangement on the chromosomes compared to the other species of grasses. So there's been quite a bit of movement of those genes from one chromosome to the other, breaking and joining chromosomes together to form either larger numbers of chromosomes or smaller numbers of chromosomes. But ultimately the genes, at least not the alleles, but the genes, are very similar in all of these. And remember, when I was talking about transposons, about 84% of the maize genome consists of transposons and retrotransposons, and 5% consists of genes, mRNA genes, and then there are introns and microRNAs, both micro and interfering RNAs. This is true of other large genomes in plants as well. And even when you get to the small genomes, a large proportion of the genome consists of transposons, and a smaller portion consists of the mRNA encoding genes. And for the maize genome, if you look at the 10 chromosomes in the maize genome, the LTR retrotransposons are located in the center of the chromosome, surrounding the centromere and going out to about the 80% mark on the chromosomes. There are intermixed DNA transposons also, and the genes are out towards the telomere ends on those chromosomes. I'm not certain of the arrangement on for the other grasses, but I suspect, since you can get synteny of the chromosomes, that the arrangement is probably similar on those chromosomes as well. And then if you look at the categories of genes in each one of those grasses compared to Arabidopsis, you find that a large number of the genes for all plants, 65 to 83%, are in common for all of those plants. So there's a central set of genes in plants, and you would find this in bacteria also, that are central to all of those organisms and all of those groups and families. But then when you focus in on just the grasses, you find another fairly high percentage are also in common among all those grasses as well. And that's seen also the fact that you can line up those chromosomes so well in the previous slides. So the next genome I want to talk about is that of Saccharomyces cerevisiae, and we discussed this briefly previously in the course. Yeasts have been used for probably 9,000 to 10,000 years in fermentation processes, both for making beer and eventually wine, and in making bread and other products, cheese and things like that. So it's been used, it's been cultivated essentially for many thousands of years, although in the beginning it was probably a mixture of yeasts rather than a pure strain of Saccharomyces like we're using today. So the genome size of Saccharomyces cerevisiae is about 12 million base pairs. And as I mentioned with the plants, this is about between 1/4 and 1/2 the size of a typical chromosome in a plant in those genomes that are smaller for Arabidopsis, for instance, or rice. 4,862 protein encoding genes. About 3% is devoted to DNA metabolism and replication, about 5% to transcription and transcription regulation, and about 13% to amino acid metabolism and translation, and then another 5% to protein modification and secretion. So there's about 6% to energy metabolism, 7% to response to stress in the environment, since it has to respond to different conditions. It's both aerobic and anaerobic. About 9% to transport and binding, that is, transport around the cell and into and out of the cell. A large proportion, 10%, to RNA metabolism. It does use RNA for signaling as well as expression control. And then 8% to the cell envelope, and cell cycle control, about 4.3%. And then there is 6%, about 6.5%, devoted to cell division, chromosome segregation, budding, and cytokinesis in those cells. So pretty complex cell division processes. And then there are small portions of the genome set aside for other functions as well. Here are the chromosomes of Saccharomyces cerevisiae. It has 16 chromosomes, And you can see the various sizes there, ranging from 230,000 base pairs, which is about the size of a large DNA virus genome, going up to chromosome 4 there with 1.5 million base pairs of DNA in that one. So that's getting to be more like a bacterial chromosome. Also note that the number of genes and proteins is pretty much the same all the way across, so there's not a lot of indication for alternative splicing in Saccharomyces cerevisiae. The tRNA genes are spread out among the chromosomes, whereas the ribosomal RNA genes are all in one locus on chromosome 12. And so if you were to electrophoresis these chromosomes on a Chef gel or something like that, you would find fairly distinct bands for 15 of the 16 chromosomes, but chromosome 12 often looks a bit fuzzy because each of the chromosomes for each of the cells has a slightly different number of ribosomal RNA genes. So those vary, so there's a little bit of fuzziness for those chromosomes. So up at the top there you can see 142 ribosomal RNA genes. That's an average, so cells would have anywhere from 100 to sometimes 200 or 250 ribosomal RNA genes. 299 tRNA genes, so quite a few tRNA genes. So each of the tRNAs then is being used at different times in different ways for different purposes as the cells respond to different external stimuli. And greater than 500 non-coding RNA genes. And so in the previous pie chart you saw there was 10% of the genome related to RNA metabolism. And this is forming a lot of these RNAs, which are controlling transcription or expression of other genes. So the next genome I want to discuss is that of Caenorhabditis elegans, which is a nematode. Or a simple animal. So it's been used extensively in genetic studies to study gene expression and development in animals. So here are a few diagrams or photos of Caenorhabditis elegans. It has two sexes, a hermaphrodite that can fertilize its own eggs, and then a smaller number of males. A fairly simple animal with a mouth, a pharynx gut, and then an anus at the back end, and then the rest of the animal is pretty much sexual organs to make the eggs and the semen in the case of the hermaphrodite, or just semen in the case of the male. It's been studied developmentally, so people have looked at the cell fates, so quite an extensive study of the cell fates in Caenorhabditis elegans. And you can find it out in your garden if you look carefully enough. Here's a pie chart of Caenorhabditis elegans genome, and it's about 100 million base pairs and has 20 to 25,000 genes. So it does have a section of its genome for DNA metabolism and replication, for transcription, for translation, etc. One of the things that has increased greatly in this multicellular animal is signal transduction. So the developmental pathway is important so that each cell in that organism has to know what it is next to, what signals it's getting from the outside, and respond to those signals. And if you have a cancer, and these guys presumably would get cancer also if you have the signal transduction pathways being inhibited or broken somehow by mutation, then those cells would just start multiplying and cause a tumor, just as they would in a human. So the main takeaway on this one is that this signal transduction set of genes has greatly expanded, and the complexity of the organism in its organization and developmental pathway have also expanded or become more elaborate. Here are the chromosomes for Caenorhabditis elegans. 5 chromosomes plus a sex chromosome, the X chromosome there. And you see the chromosome sizes ranges from about 14 million base pairs up to almost 21 million base pairs. You should note on this one now that the number of genes, well, it's around 25,000 genes. The number of proteins that can be formed is more than 26,000. And so here in these animals, you're starting to get glimpses of extensive alternative splicing so that one gene can form more than a single protein by splicing the exons in different ways. Also, there's the average number of ribosomal RNA genes at 275, although again, this varies from one cell to the next. 590 tRNA genes and 120 microRNA genes, which are controlling expression of other RNAs. 70 spliceosomal RNA genes, which are responsible for the splicing and alternative splicing of those genes, and then 30 SNO RNA genes, which are primarily responsible for processing of the ribosomal RNAs, although they have been found to be part of the process of processing other RNAs as well. So I hope you're seeing a pattern now that as you have more and more elaborate organisms, although there are essentially more genes in general, there's more alternative splicing taking place to increase the number of protein products from those genes, and there's a lot more small non-coding RNAs that are causing differences in expression of the mRNAs from those genes. The next genome I want to discuss is that of Drosophila melanogaster, or fruit fly. It's been used, this organism's been used for many decades as a genetic model. So in this case, the developmental pathway is getting more complex than that of Caenorhabditis elegans, the nematode. So starting from the oocyte, there's already differences from one end of the egg to the other in the proteins and the expression of RNAs. And then fertilization occurs. You have a nuclear syncytium, where you have nuclear division without cellular division. And then finally, the formation of the blastoderm, where the nuclei move out to the edge and do form cells. And then expression of different developmental regulation genes, the gap genes, pair rule genes, and polarity genes, to set up the segmentation of the organism. And then overlaid on top of that are the homeotic genes, which take those segments and determine what sorts of organs to place in those positions. Now, most of these, the gap genes, the pair rule genes, polarity genes, homeotic genes, are transcription factors that are influencing the transcription of many other genes to set up those patterns. So here's the genome of Drosophila melanogaster. You have 2L and 2R, that is the left and right arms of chromosome 2, and then the right and left arms of chromosome 3, and then chromosome 4 and the X chromosome. And each one of those has from about 13.5 million base pairs to close to 28 million base pairs on them. So they're getting pretty large. And then you can see, as you saw in Caenorhabditis elegans, that there are more proteins than there are genes, indicating again alternative splicing. Also, between 100 and 240 ribosomal RNA genes. So that's again the same pattern we've seen where there's a variable number from cell to cell. 297 tRNA genes, and now 15,000 non-coding RNA genes. So more and more control from those small RNAs that are controlling expression of the already produced mRNAs usually. So when we were talking about Drosophila melanogaster development, that is animal development, we talked about some of these products and how they influence the expression of other products and genes. So up here, NOS inhibits BCD, and BCD inhibits CAD, and so you have a difference in the concentrations of each one of those, including HB, which is hunchback. And then going down to the GAP genes, these products up here can affect turning on and off of those genes. And you go down further to the parallel genes, those are influenced by the products of the previous genes and products that were turned on. And finally, the segment polarity genes down here are influenced by these genes up here. And eventually you have a segmentation and finer segmentation along the length of the animal. And humans have this also. If you look at the vertebrae, you have segmentation in that way as well. And then you get down to the homeotic genes, which will then cause the placement of certain organs in different areas of the organism based on their influence on turning on and off genes that will form those different appendages and organs. So in general, while there are more genes in each one of these genomes as you get to more and more complex organisms, one of the main things you see is more gene expression control through transduction pathways, through microRNAs, through alternative splicing to fine-tune each one of the cells, cell types, and tissues within those organisms. And as you work your way from fairly simple animals, sea urchins, through nematodes, Drosophila, fish, amphibians, and mammals, you find that these mechanisms are similar in all of these, but how they're turned on and the number of genes that are turned on and the transcriptional control vary, so that they all have similarities. And you can see a definite developmental and evolutionary pattern. They also have differences that reflect in the different body types and complexities of those organisms. On the left here, you also see how they're turned on in a normal Drosophila and in a mutant, and so that in the normal you get the expected segmentation, whereas in the mutant, where you've just mutated one or a few of those genes, you have a very different pattern. And normally, if this happened in an organism, it would abort before it fully matured. And some of these mutations are fairly common, and even in human beings, you have a lot of spontaneous abortion, and some of those are caused by mutations in these genes that are acting very early in development, so that the embryo and fetus aren't formed accurately. And you can see as you work your way through the different animals also the differences in these Hox genes. And so if you have a mutation, for instance, in those anterior genes, that would tend to affect organs towards the head end of those organisms. Whereas if you have mutations on the right-hand side of this, on the 5' side, or 5' side of those gene loci, you would find more mutations in the appendages on those organisms, or in the abdomen in the case of fruit flies and some of the insects. And again, these genes themselves and these gene products are affecting transcription of many, many other genes, so that their effects can be very wide-ranging, and they can affect many different cell types. Next, I want to talk about Homo sapiens, which has approximately 23,000 to 27,000 genes, depending on how people are counting them. This is a species that's quite variable morphologically and spread throughout the world. Almost 8 billion humans right now in the world. And if they're also hybrids, so that, most are hybrids, so that they have hybridized with other species in the genus Homo. So they're not uniform either, although they can all reproduce. One of the reasons we know that humans have hybridized is that the genomic sequences of many humans have been determined. And it's not the entire genomes. Usually they're looking at specific loci and looking also at single nucleotide polymorphisms, SNPs, within the different populations. The partial sequences of Neanderthals and Denisovians have also been done, although the Denisovians, as far as sequence, is really not nearly complete at this moment. But we do know that there were hybridization events in Africa that have happened, possibly with Homo erectus, although one group thinks it's another group of Neanderthals that moved back into Africa. And then there were hybridization events in the Arabian Peninsula with Neanderthals, and another possible one up in Europe. These hybrids survived because they were selected for. So the immune systems of the Neanderthals were attuned to different regions, and when the humans coming out of Africa bred with them, those factors, especially the immune system and avoiding pathogens, or at least expunging pathogens, was a distinct advantage for those hybrid species. There was another hybridization event in Asia, possibly with the Denisovians, and there may have been another one with Homo erectus over in that same area. And then the humans spread out throughout the world, including moving over into North America and South America in the past 14,000 years. And then another group, the Inuits, moving more recently into the Arctic region of North America as well. Here are the chromosomes of Homo sapiens laid out. The smallest chromosome has about 33.5 million base pairs of DNA, while the largest has over 250 million base pairs of DNA. And there are the number of genes on each one of the chromosomes. And you can see here at the end 29,841 genes in this case, although I've seen some studies where it's as low as 23,000 genes. And we can make about 91,000 proteins. So a lot of alternative splicing. In some cases, a single gene can make 6 or 7 different proteins. And that's the case in the dystrophin gene. Also, there are from 150 to over 2,000 ribosomal RNA genes, again indicating the amount of variation in the ribosomal RNA genes, which we've already spent a lot of time talking about. 497 tRNA genes, 97 SNO RNA genes, which are responsible for processing ribosomal RNAs, as well as other RNAs, and a large number of spliceosomal RNA genes for that alternative splicing. So again, in this genome, as we've seen with other genomes where you have elaborate multicellular organisms, a lot of alternative splicing happening and a lot of control by small RNAs, and I haven't listed all the microRNAs in humans also, but there are a lot of those as well. Another large portion of the genome consists of transposons and retrotransposons, And of course, these are responsible for a lot of exchanges in among the genes. So this will increase the evolution of already existing genes. About 1.5 to 2% of the genome is made up of mRNA genes, those that encode proteins, and about 10% of the genome is made up of non-coding RNA genes. So again, more control of expression of already formed RNAs in this case. And then 48% of the genome is relatively unknown, although a lot of that consists of introns. There are some introns that are over 1 million base pairs in length. So just to summarize a bit, then if you look at all animals and fungi, which are all opisthecons, then 25% of their genomes, their genes are in common, so that you have a basic set of genes that they all have, and then you elaborate on that. So another 43% are common among all of the animals, and then you get these unique sets of genes that that then elaborate each one of those organisms as far as their body plan and their final look and what they can do and how they reproduce, and etc. Also, if you lay out their chromosomes side by side, you find that although the chromosome set and the number of chromosomes, the size of the chromosomes vary by quite a bit, you can line up different sections of those. So between humans and mice, you can see that chromosome 1 in humans, part of that exists on chromosome 1 of mice and chromosome 4 in mice. And then as far as chromosome 2, parts of that are on chromosome 1 in the mouse, and then chromosome 2 in the mouse genome, and in other places chromosome 6. And so although they have the same genes, they're laid out in different places, and that can affect somewhat the expression of those genes as well, so that then you get a human instead of a mouse. It also provides some barrier so that you can't make a mouse-human hybrid, because the genes on the chromosomes simply don't line up properly. Also, as organisms become more and more complex, you find that they have more transcription factors. So I haven't listed the ABCD, the type of transcription factors, but these are all transcription factors, and you can see as you go from Saccharomyces, which has smaller numbers of these transcription factors for each category. Cinerobditis has more, Drosophila melanogaster has more, Homo sapiens have more also. So these genes that encode transcription factors are in increasing numbers as those organisms become more complex, and their developmental pattern becomes more complex as well. But remember, the complexity of an organism is not necessarily related to its genome size per se, so that in the case of some protozoans, they have much larger genomes, although their complexity is lower. That is, their developmental pattern and whether they're unicellular or multicellular is much more simple than, for instance, mammals, birds, dinosaurs. So there are some things that are directly related to genome size, but more directly related to the number of proteins, the amount of transcription factors, the number or the amount of alternative splicing. Those all relate to more complexity in the organism. Also, unicellular organisms that live in the environment, in many different environments, have slightly larger genomes. Those that exist only in limited environments have smaller genomes, and then those that become symbionts, endosymbionts, have smaller and smaller genomes. And then as you work your way up into multicellular organisms, they have to have more control because those cells have to work in a coordinated way. So they have more control over the genome, and that's usually by increasing the number of proteins and genes that are involved in signal transduction, control of the cell cycle, recognizing DNA damage, etc. So there are several parts of evolution. The first part is being just simple mutation within a genome. But then you can have interaction between two different species, or the same two individuals of the same species, through the process of horizontal gene transfer. And in that way, you can speed up essentially evolution by moving genes from one organism into the other and then sorting those out by selection. The next thing that can happen is you can have endosymbiotic events. Well, first mutualistic events and parasitic events where you move around larger portions of the genome. And then you can have endosymbiotic events where you essentially join two fairly large genomes together into a single organism. And these have been characteristic of eukaryotes. So both taking in a bacterium into a eukaryote to become a mitochondrion and a plastid or a chromatophore, but also taking in a eukaryote into a eukaryote cell, such as a red alga or a green alga, being taken up by a protist cell, thus making it, initially anyway, a photosynthetic organism. And then you have the mixing of those genomes, and that greatly speeds the rate of evolution because there's initially a sorting out of the specific genes that are going to allow that organism to be successful. So in this figure I've shown some of those events, but on this diagram, this phylogram, I've superimposed some of those endosymbiotic events. So here's the mitochondrion from the alpha proteobacterium, the cyanobacteria entering the plants to become red and green algae, and then another event in the rhizaria to become pollinella, and then red and green algae entering the excavata, alveolata, Hacrobia, Stramenopile clades to become other organelles. And you get all of these genomes joining together and functioning in sometimes those chimeric pathways I was talking about, which lead to further expansion in those groups and different types and speeded evolution in some cases. So this leads then to these rather large genomes that you see in many cases in protozoans and plants, and also gives you a great deal more in diversity in each one of those groups. And many, as I've stated before, many of those organisms that once were photosynthetic have become parasitic. So then you have a different branch in those groups that lose their photoautotrophy in favor of being parasites, and that changes the evolution on that line of that group as well. Okay, so that's the end of this presentation on eukaryotic genomes. As I said, I covered them in a little less detail than I did way back with the viruses, but all of the things and mechanisms, molecular mechanisms that you see in viruses and bacteria are also happening in eukaryotes, but to a much larger extent. Also in eukaryotes, they're really combinations of cells, so it's genomes interacting with other genomes, and then evolutionary processes that are sort of weeding out or sorting out the individual duplications and repetitions of various genes and pathways. Okay, well, thank you very much, and I will talk to you later.